Welcome to my YouTube channel. In this powerful and insightful video, here we discuss some key points. Discover the fundamental chemistry concepts behind antibiotics. Antibiotics are drugs used to treat bacterial infections. They work by targeting specific structures or processes within bacteria to inhibit their growth or kill them. The fundamental chemistry concepts behind antibiotics involve understanding the chemical structures of these drugs and how they interact with bacterial cells. 1. Molecular structure. Antibiotics are organic compounds that have specific functional groups and chemical structures. Their structures are designed to interact with bacterial cell components in a way that disrupts their normal function. Understanding the molecular structure of antibiotics is crucial for developing new drugs with improved efficacy and reduced side effects. 2. Targeting bacterial cell walls. Some antibiotics, such as penicillin and cephalosporins, target bacterial cell walls. Bacterial cell walls are primarily composed of peptidoglycan, a unique polymer made up of sugar molecules cross-linked by short peptide chains. Penicillins and cephalosporins inhibit the enzymes involved in peptidoglycan synthesis, leading to the weakening and rupture of the bacterial cell wall. This causes the bacterium to burst, leading to its death. 3. Interfering with protein synthesis. Many antibiotics, such as tetracyclines, macrolides, and aminoglycosides, disrupt bacterial protein synthesis. These antibiotics target the ribosomes, the cellular structures responsible for translating the genetic code into functional proteins. By binding to specific regions of the ribosome, these antibiotics prevent the synthesis of essential proteins, leading to the inhibition of bacterial growth and survival. 4. Inhibiting enzyme activity. Certain antibiotics, like sulfonamides and trimethoprim, inhibit the activity of enzymes that are crucial for bacterial survival. These antibiotics target enzymes involved in processes such as folic acid synthesis, nucleic acid synthesis, and metabolic pathways. By inhibiting these essential enzymes, antibiotics disrupt the normal functioning of bacterial cells, ultimately leading to their death. 5. Blocking DNA replication. Some antibiotics target bacterial DNA replication, preventing the bacterium from reproducing effectively. Drugs like fluoroquinolones inhibit the activity of bacterial DNA gyrase and topoisomerase IV enzymes, which are essential for DNA replication and repair. By interfering with these enzymes, these antibiotics disrupt the bacterial DNA structure and prevent replication, leading to bacterial cell death. Understanding the fundamental chemistry concepts behind antibiotics is crucial for developing new drugs, improving existing ones, and combating the emergence of antibiotic resistance. By targeting specific bacterial processes and structures, antibiotics effectively treat infections and save countless lives. 6. Disrupting cell membranes. Another mechanism of action for certain antibiotics is the disruption of bacterial cell membranes. Polymyxins, for example, interact with the lipids in the bacterial cell membrane, causing destabilization and leakage of cellular contents. This disruption compromises the integrity and functionality of the bacterial cell, leading to its death. 7. Generating reactive oxygen species. Some antibiotics, such as quinolones, can generate reactive oxygen species, ROS, within bacterial cells. ROS are highly reactive molecules that can cause significant damage to cellular components, including DNA, proteins, and lipids. Accumulation of ROS overwhelms the bacterial cell's defense mechanisms and leads to oxidative stress, eventually resulting in cell death. 8. Altering bacterial metabolism. Antibiotics like sulfonamides and trimethoprim can interfere with bacterial metabolism. These drugs target specific enzymes involved in the synthesis of essential metabolites, such as nucleotides or vitamins. By inhibiting these enzymes, antibiotics disrupt the production of vital cellular components, hindering bacterial growth and survival. 9. Overcoming resistance. Over time, bacteria can develop resistance to antibiotics through various mechanisms. Efflux pumps, enzymatic inactivation, target modification, and altered membrane permeability are examples of resistance mechanisms. Understanding the chemical underpinnings of antibiotic resistance is crucial for developing strategies to overcome it. This involves designing new antibiotics or modifying existing ones to evade resistance mechanisms and preserve their effectiveness. 10. Pharmacokinetics and pharmacodynamics. Antibiotics also require an understanding of pharmacokinetics and pharmacodynamics. Pharmacokinetics involves the study of drug absorption, distribution, metabolism, and elimination within the body. Pharmacodynamics focuses on the drug's interactions with its target site, including its concentration, time-dependent or concentration-dependent effects, and efficacy against the pathogen. These concepts help determine optimal dosing regimens, combination therapies, and strategies to minimize side effects. Overall, the fundamental chemistry concepts behind antibiotics encompass a range of mechanisms by which these drugs target and disrupt bacterial cells. These concepts include molecular structure, target interaction, enzyme inhibition, DNA interference, and metabolic disruption. By understanding these principles, scientists can continue to develop new antibiotics and combat the ever-evolving nature of bacterial infections. Learn about the different classes and mechanisms of action of antibiotics. Antibiotics are a class of drugs used to treat bacterial infections. They work by inhibiting the growth or killing bacteria. There are various classes of antibiotics, each with a different mechanism of action. Understanding these classes can help in understanding how antibiotics work and in choosing the right antibiotic for a specific infection. 1. Beta-lactams. This is the largest class of antibiotics and includes penicillins, cephalosporins, carbapenems, and monobactams. Beta-lactam antibiotics work by inhibiting bacterial cell wall synthesis. They prevent the cross-linking of peptidoglycan, a major component of the bacterial cell wall, leading to cell wall instability and bacterial death. 2. Macrolides. Macrolides, such as erythromycin and azithromycin, work by inhibiting bacterial protein synthesis. They bind to the bacterial ribosome and prevent the elongation of the growing protein chain. 3. Tetracyclines. Tetracyclines, including doxycycline and minocycline, also inhibit bacterial protein synthesis. They do so by binding to the bacterial ribosome and blocking the attachment of aminoacyl tRNA molecules. 4. Aminoglycosides. Aminoglycosides like gentamicin and streptomycin interfere with bacterial protein synthesis. They bind to the bacterial ribosome and cause misreading of the genetic code, resulting in the production of non-functional proteins. 5. Quinolones. Quinolones, such as ciprofloxacin and levofloxacin, interfere with bacterial DNA replication and repair. They target the bacterial enzymes called DNA gyrase and topoisomerase IV, which are involved in DNA synthesis. 6. Sulfonamides. Sulfonamides, such as sulfamethoxazole, inhibit bacterial folic acid synthesis. They act as competitive inhibitors of the enzyme dihydrooctanoate synthase, which is involved in the synthesis of folic acid, an essential nutrient for bacterial growth. 7. Glycopeptides. Glycopeptides, like vancomycin, work by inhibiting bacterial cell wall synthesis. They bind to the building blocks of the bacterial cell wall and prevent its cross-linking, leading to cell wall destabilization. These are just a few examples of the different classes of antibiotics and their mechanisms of action. It's important to note that bacteria can develop resistance to antibiotics over time, which is why it is crucial to use antibiotics judiciously and follow proper treatment guidelines to combat antimicrobial resistance. Certainly. Here are a few more classes of antibiotics, each with distinct mechanisms of action. 8. Oxazolidinones. Oxazolidinones, such as linezolid, inhibit bacterial protein synthesis. They bind to the 50S ribosomal subunit and prevent the formation of the initiation complex, thereby blocking the production of proteins. 9. Lipopeptides. Lipopeptides, like daptomycin, act by disrupting bacterial cell membrane function. They bind to the bacterial membrane and cause depolarization, leading to cell death. 10. Rifamycins. Rifamycins, such as rifampicin, inhibit bacterial RNA synthesis. They bind to the bacterial RNA polymerase and prevent the synthesis of RNA, thereby disrupting bacterial gene expression. 11. Nitroimidazoles. Nitroimidazoles, including metronidazole, have a unique mechanism of action. They are activated by the reduction of nitro groups within bacterial cells. The resulting compounds interfere with bacterial DNA synthesis and cause DNA damage, leading to bacterial cell death. 12. Polymyxins. Polymyxins, such as polymyxin B and colistin, disrupt bacterial cell membranes. They bind to the lipopolysaccharides in the outer membrane of gram-negative bacteria, causing damage and ultimately leading to cell death. 13. Lincosamides. Lincosamides, like clindamycin, inhibit bacterial protein synthesis. They bind to the 50S ribosomal subunit and prevent the formation of peptide bonds during protein synthesis. 14. Folate pathway antagonists. Antibiotics like trimethoprim and sulfamethoxazole inhibit bacterial folic acid metabolism by targeting different enzymes of the pathway. Trimethoprim inhibits dihydrofolate reductase, while sulfamethoxazole inhibits dihydropteroate synthase. These are just a few additional classes of antibiotics and their mechanisms of action. It's important to note that the choice of antibiotic depends on several factors, including the type of infection, the causative bacteria, and the antibiotic spectrum of activity. Proper use, dosage, and duration of antibiotic treatment are essential to ensure effectiveness and minimize the development of antibiotic resistance. Gain insights into how antibiotics target and inhibit bacterial growth. Antibiotics are powerful medications used to treat bacterial infections by targeting and inhibiting the growth of bacteria. They work through various mechanisms, which can be broadly categorized into four main types: inhibition of cell wall synthesis, inhibition of protein synthesis, disruption of nucleic acid synthesis, and inhibition of essential metabolic pathways. 1. Inhibition of cell wall synthesis. Many bacteria have a rigid cell wall that provides structure and protection. Antibiotics targeting cell wall synthesis interfere with the construction of this protective barrier, causing the bacteria to become more susceptible to damage. The most well-known antibiotics in this category are the beta-lactam antibiotics, such as penicillins and cephalosporins, which inhibit the formation of peptidoglycan, an essential component of the cell wall. By inhibiting cell wall synthesis, these antibiotics weaken the bacteria and can ultimately lead to cell lysis. 2. Inhibition of protein synthesis. Proteins are essential for bacterial growth and replication. Antibiotics targeting protein synthesis interfere with the machinery responsible for translating the genetic information into functional proteins. These antibiotics can bind to ribosomes, the cellular structures responsible for protein synthesis, and disrupt the process by blocking the initiation, elongation, or termination steps. Examples of antibiotics targeting protein synthesis include macrolides, e.g., erythromycin, aminoglycosides, e.g., gentamicin, and tetracyclines. 3. Disruption of nucleic acid synthesis. Nucleic acids, including DNA and RNA, contain genetic information necessary for bacterial survival and replication. Antibiotics that inhibit nucleic acid synthesis interfere with the replication and transcription of bacterial genetic material, preventing further growth and multiplication. Quinolones, e.g., ciprofloxacin, and rifampin are examples of antibiotics that disrupt nucleic acid synthesis. Quinolones interfere with the enzymes responsible for DNA replication and repair, while rifampin inhibits the bacterial RNA polymerase. 4. Inhibition of essential metabolic pathways. Various metabolic pathways are crucial for the survival and growth of bacteria. Antibiotics targeting these pathways disrupt key metabolic reactions, leading to the impairment of bacterial function. For instance, sulfonamides and trimethoprim inhibit the synthesis of folic acid, a vital molecule required for the synthesis of nucleic acids and certain amino acids. By blocking folic acid production, these antibiotics cripple bacterial metabolism and impede further growth. It's important to note that antibiotics are designed to specifically target bacterial cells while minimizing harm to the host organism cells. This selectivity is possible due to differences in the cellular structures and biological processes between bacteria and humans. However, it is worth mentioning that bacteria can develop resistance to antibiotics over time. This can occur through various mechanisms, such as genetic mutations or the acquisition of resistance genes from other bacteria. The misuse or overuse of antibiotics can accelerate the development of antibiotic resistance, posing a significant challenge in modern healthcare. Therefore, it is crucial to use antibiotics responsibly, according to healthcare professionals' guidance, to preserve their effectiveness. Certainly. Here's some additional information on how antibiotics target and inhibitory growth. 5. Disruption of cell membrane. Some antibiotics target the bacterial cell membrane, which is responsible for maintaining cell integrity and controlling the transport of molecules. Polymyxins, for example, are antibiotics that interact with the lipid components of the cell membrane, leading to destabilization and leakage of cellular contents. By disrupting the cell membrane, these antibiotics disrupt bacterial homeostasis and inhibit growth. 6. Enzyme inhibition. Certain antibiotics inhibit specific enzymes that are essential for bacterial growth and survival. For instance, sulfonamides are structural analogues of the compound para-aminobenzoic acid, PABA, which is required for the synthesis of folic acid. Sulfonamides competitively inhibit the enzyme that converts PABA to dihydrofolic acid, preventing the production of folic acid and impeding bacterial. 7. Targeting essential cellular processes. Other antibiotics target specific cellular processes that are vital for bacteria but absent or different in humans. For example, antibiotics like daptomycin and cyclic lipopeptides disrupt bacterial cell membrane integrity by creating ion channels, depolarizing the membrane, and ultimately causing cell death. Since human cells lack similar structures, these antibiotics have a selective effect on bacteria. It's important to note that different classes of antibiotics have different mechanisms of action and target different aspects of bacterial growth. Therefore, a combination of antibiotics may be prescribed in severe or complicated infections to maximize effectiveness and combat the development of resistance. Additionally, it's worth highlighting that antibiotics have no effect on viral infections since viruses have different structures and replication mechanisms compared to bacteria. Antibiotics are specifically designed to target bacterial cells and are ineffective against viruses such as the common cold or influenza. To ensure the optimal use of antibiotics and prevent the spread of antibiotic resistance, it is crucial to follow proper prescribing guidelines, complete the full course of medication as prescribed, avoid unnecessary antibiotic use, and practice good infection prevention and control measures. In summary, antibiotics exert their inhibitory effects on bacteria by targeting various crucial components, such as the cell wall, protein synthesis machinery, nucleic acid synthesis, metabolic pathways, cell membrane, and essential enzymes. Understanding these mechanisms helps scientists and healthcare professionals develop effective antibiotics and combat bacterial infections. Understand the importance of antibiotic stewardship and avoiding misuse. Antibiotic stewardship and avoiding the misuse of antibiotics are crucial for several reasons. Let's explore their importance. 1. Preservation of antibiotic effectiveness. Overuse and misuse of antibiotics contribute to the development of antibiotic resistance. When antibiotics are used unnecessarily or inappropriately, bacteria can adapt and become resistant to these medications, rendering them ineffective in treating infections. Antibiotic resistance is a global health concern as it limits our ability to effectively treat bacterial infections, leading to longer and more severe illnesses. 2. Improved patient outcomes. Antibiotic stewardship programs aim to optimize patient outcomes by ensuring the right antibiotic is prescribed, at the right dose, for the right duration. When antibiotics are used appropriately, infections are more likely to be successfully treated, reducing complications and improving patient recovery. 3. Minimization of adverse effects. Antibiotics can have side effects such as nausea, diarrhea, allergic reactions, and the potential for more severe adverse events. By avoiding unnecessary antibiotic use, the risk of experiencing these side effects is reduced. 4. Cost reduction. Inappropriate use of antibiotics can contribute to unnecessary healthcare costs. By avoiding unnecessary antibiotic prescriptions, healthcare systems can allocate resources more effectively, reducing financial burden. 5. Preservation of broad-spectrum antibiotics. Certain antibiotics are considered broad-spectrum, meaning they can target a wide range of bacteria. These antibiotics are often reserved for more severe infections or cases where the specific bacteria causing the infection is unknown. By avoiding their misuse, we can help preserve the effectiveness of these important medications for when they are truly needed. 6. Prevention of Clostridium difficile infections. Misuse of antibiotics can disrupt the balance of bacteria in the gut, leading to an overgrowth of the bacteria Clostridium difficile. This can result in severe and recurrent infections known as C. difficile infections. By using antibiotics judiciously, the risk of developing such infections can be minimized. To promote antibiotic stewardship and avoid the misuse of antibiotics, it is essential for healthcare providers, patients, and the general public to understand the appropriate indications for antibiotic use, complete prescribed courses, and adhere to evidence-based guidelines. Additionally, explore the future of antibiotics and the challenges we face in combating antibiotic resistance. Antibiotics have saved countless lives since their discovery, but the rise of antibiotic resistance poses a significant threat to public health in the future. Antibiotic resistance occurs when bacteria evolve mechanisms to survive and grow in the presence of antibiotics, rendering these drugs less effective or completely ineffective. The future of antibiotics involves addressing several key challenges. One challenge is the overuse and misuse of antibiotics. Antibiotics are commonly prescribed for viral infections where they have no effect, or used inappropriately in agriculture, leading to increased antibiotic resistance. Another challenge is the slow pace of antibiotic development. Discovering and developing new antibiotics is a complex and time-consuming process. Many pharmaceutical companies have shifted their focus away from antibiotic research due to financial and regulatory challenges, leaving us with a limited pipeline of new drugs. Additionally, the natural process of bacterial evolution allows for the rapid development and spread of antibiotic resistance. Bacteria can acquire resistance genes through mutation or by exchanging genetic material with other bacteria, including those of different species. These genetic changes can occur quickly and make bacteria that were once susceptible to antibiotics resistant, making it difficult to stay ahead of the evolving microbial. To combat these challenges, various strategies are being pursued. One approach is improving the responsible use of antibiotics through better diagnostics and guidelines. Rapid diagnostic tests can help doctors determine whether an infection is caused by a bacterium that requires antibiotic treatment, reducing unnecessary prescriptions. Enhanced stewardship programs promote appropriate antibiotic use and educate healthcare professionals and the public about the risks of antibiotic resistance. Another strategy involves developing alternative treatments to antibiotics. These include targeted therapies such as phage therapy, where specific viruses, bacteriophages, are used to infect and kill harmful bacteria, and antimicrobial peptides derived from natural sources. There is also ongoing research into novel classes of antibiotics, such as those targeting different bacterial processes or utilizing combinations of drugs to overcome resistance mechanisms. Advanced technologies like genomics and synthetic biology offer promising tools for studying and combating antibiotic resistance. They can help identify new targets for drug development, understand the mechanisms of resistance, create new molecules, and design more effective treatment approaches. Options. By doing so, we can extend the effectiveness of current antibiotics and ensure that we have new solutions to combat antibiotic-resistant infections, safeguarding public health for generations to come. In this video, we delve into the fascinating world of antibiotics chemistry. We unravel the chemical structures and mechanisms of these essential medications, shedding light on how they combat bacterial infections. From the discovery of penicillin to the latest advancements in antibiotic development, this video provides a comprehensive overview of antibiotic chemistry. We discuss the different classes of antibiotics, including beta-lactams, tetracyclines, fluoroquinolones, and more, highlighting their unique chemical properties and how they target specific bacterial components. Furthermore, we explore the mechanisms by which antibiotics disrupt bacterial growth, such as inhibiting cell wall synthesis, protein synthesis, or DNA replication. Understanding the chemistry of antibiotics is crucial for appreciating their effectiveness and limitations. It allows us to comprehend the importance of appropriate antibiotic use, as well as the potential consequences of misuse or overuse. By advocating for antibiotic stewardship, we can preserve the efficacy of these life-saving medications for future generations. Join us in this educational journey to gain a deeper understanding of antibiotics chemistry and their role in healthcare. Together, let's combat antibiotic resistance and ensure a sustainable future for these invaluable drugs. If you found this video informative, please consider subscribing to our channel for more educational content on various scientific topics.